this, and we'll go further here. So Rothman had his postdocs, and they purified from the CHO 15B and the CHO. And again, the CHO 15 lacked the Golgi NAG transferase. They cannot make complex sugar trees. So acceptor membranes were from uninfected CHO cells. So they had the donor membrane, the CHO 15 cis, to the acceptor membrane, the uninfected wild type CHO cells. And what does the virus look like here? Let me pause. Gotta have some more water. Hey guys, we're back. Got a little water, cough drop here. So I wanted to kind of give you an idea as we're getting into the virus, exactly what the virus is. Okay, so this is the structure of the virus. It's kind of related to the rabies virus. It's taken into the cell, the endosome and the lysosome. It becomes acidic, and that allows the virus nucleocapsid to break in, and eventually we end up getting the genome. And this is the annotation of the virus genome. There's the N, the P, the M, the G, and the L genes. And this is the G protein right here. So they're just showing you the G protein. So the virus is making G protein, and the G protein, of course, goes into the lumen of the R. And so then, just to remind you, it's decorated with a sugar tree, the oligosaccharide, and there's some enzymes that chew off glucose and one of the mannoses. And then in the cis Golgi, there's the first enzyme, the glutamac transferase 1, that removes the mannose, and you end up getting a sugar attached to it. And if you don't have that, then none of these other reactions occur. So in the CHO 15, they don't have this. So that means it will never mature past this point right here. Stays right there. It will not make a complex oligosaccharide. Okay, so they took the CHO 15 cells and infected them with pure virus, got the G protein, and then probably after the infection, they cooled it down to 4 degrees, did a two-step fractionation, subcellular sucrose density fractionation we've talked about, purified these stacks from the CHO 15. At the same time, they had pure Golgi stacks from the CHO wild type. And just to show you, if you tag the G protein, you can see it's in the ER, and by 40 minutes, it's pretty much in the Golgi, and then through about 180 minutes, it's out. Some still in the Golgi, but it's moving back out. So ER Golgi plasma membrane here, and you can actually get the speed here, and it just shows you over in time of infection. So it's high in the ER, everything leaves the ER, then the G protein hits the Golgi, and then eventually leaves the Golgi, and you'll start seeing the protein on the plasma membrane. And some of the proteins degraded over time. So just wanted you to get a feel for the virus, the G protein, what's it doing. And then previously we talked about the mutant 15B cells and the CHO wild type. and the carbohydrate decoration. So if you don't, you'll need to get all those concepts if you need to explain this experiment and the significance of it. So this is a probably the most complex, uh, detailed issue that or block of material we've discussed. Okay, so here's another cartoon showing the Bosch experiment. And again, Rothman, it was his lab, so he got the Nobel Prize with these guys. Okay, Bosch, Rothman. So here they got the donor Golgi fraction from virally infected, and see, they don't say CHO, CHO cells, CHO 15 Newton. It doesn't have the glutamac, so it's frozen right there. It will not, no further trimming will occur. So if you mix the acceptor Golgi from uninfected, okay, so that's the key. These CHO cells were not infected, but they do have the wild type Golgi stack. If you mix these together and the Golgi recognize self, then this experiment would have never worked. In other words, this would have just gone there and eventually to the trans and then out, and this would have had no G protein to begin with. But it did work. So vesicles, and they're not showing the vesicles, were transported and they fused with the acceptor compartment. So the cis donor to the medial acceptor compartment in the presence of ATP and cytosol. You're saying, well, how do we prove that ATP is required? Don't add it in a cell-free system, and you would never find on the G protein the NAG sugar. They're saying, well, how do we know the NAG? They radio labeled it with tritium. It's a radio isotope, and as it breaks down, it releases energy, so you can actually count the energy, the radioactive energy. If you purify the G protein, you can figure out how much G protein was radioactive because it had made it to this compartment. See, it's UDP tritium, that's an isotope, glutamac, ATP, and cytosol. So they had then reconstructed a transport from the cis to the medial, from a donor to the acceptor, in the presence of all the proteins in the cytosol and ATP as an energy source. And you could incubate for various times and see how efficient this is. Okay, you can measure that. You could purify the G protein and measure how much was radioactive over time. So you could plot time versus amount of radioactive G protein, okay, over time. This is the amount of radiation, the G protein that's radiated here, okay, radioactive over time in their system. Transport of a protein between Golgi compartments. They had invented a cell-free system to study this process. Here's just another cartoon, same concept, donor and acceptor. Shh, shh, no, shh, hey, okay. And you can read the words. Basically, it's what I've told you, but they actually have pictures of these fractions. They added cytosol and ATP for 15 minutes at 0 degrees. That froze everything. Or they did the same mixture at 37 degrees. And what did they find? They find that 0 degrees, which is here, 0 degrees, notice there's no vesicles with the Golgi. At 37 body temperature in their test tube, what do they see? Vesicles formed and moving to the next stack. Okay, vesicles. So guess what? They could purify those vesicles by more fractionation. And so not only can we study the process, but now we can study the vesicles that are doing the process. That's why the Nobel Prize, because this gave us an understanding of vesicle transport. How does the cell move material from organelles? Well, we can purify the vesicles. We can heat them at 37 and then fractionate them again, and these vesicles will be isolated. Okay. Another important thing they found was they found an inhibitor that if they added to the cytosol would, even at 37, would shut down vesicles, called the NEM inhibitor, and it was back on the other slide. For whatever reason, it won't let me go back, but that isolated one of the key factors involved in vesicle transport is called the NEM sensitive factor, NSF. And so that's my 10-cent tour of Balch's key experiment from Rothman's lab. By the way, Balch went on to do cell-free ER to Golgi stacks. Okay, so he stepped it further back and studied leaving the ER. What did it take to get into the ER? What are the proteins? And leave the ER to hit the Golgi. So, and then of course you could study leaving the Golgi to the lysosome, the endosome, the plasma membrane, the secretory granules. This set the table for all these other experiments. That's why it's the experiment to study. Okay. Wow, that took longer than I thought. Okay, so we will. I'm gonna upload this. May have to break it into two hunks here. Okay, go back and review the abstract. Go back and review the virus. Go back and review the glycosylation. You're gonna have to master all this for you to write a couple pages on this experiment. Okay, so if you know, details are your friend. That's what I'd be working on. But this is something you're just going to have to step up and figure out. Okay, we'll talk about protein fractionation coming up.